So here I'm showing different types of analytical platforms that are important when it comes to characterizing your bioprocess workflow, but also your clinical or commercial product. So it's important to appreciate that it's not possible to develop a process with only one analytical method. You need a number of orthogonal analytical methods to understand key aspects of your process. So, for example, it's important to understand and determine your capsid number. And typically, this is done using ELISA, but it's also important to understand the proportion of capsids that are full. And typically, this is done using qPCR or ddPCR, which is more accurate when it comes to quantifying viral genome concentrations, and i.e., the capsids that are full. So, over the next few slides, I'm going to look at the importance of determining capsid titer and how the ELA platform has really been instrumental to our process development. So, as I mentioned, ELISAs are commonly used for physical titer quantification. And in bioprocessing, they are widely used to understand product concentration to optimize the process, but also to look at recovery across each unit operation. ELISAs have also been traditionally used because they're quite good at dealing with impure or low concentration samples, which other analytical platforms, such as gel-based techniques, would struggle with. So, although there are a number of advantages to the ELISA, there are also a number of challenges when it comes to carrying out an ELISA. So, one of which I've demonstrated here with the schematic, and this encompasses extensive hands-on time and limited throughput. So we all know it takes roughly 5 to 7 hours to complete a traditional ELISA. So that's an entire day to get data back. And you may require data quite quickly as you're making in-process related decisions. An ELISA also has quite a narrow dynamic range. And typically, this means you have to run a number of dilutions for your sample, again, limited in the amount of throughput. And here I'm just showing a schematic of an ELISA plate map representation. And you can see a number of dilutions are required. And actually, if we use the example of 800 samples, this would mean you need to run 80 to 100 plates to achieve analysis of those 800 samples. So overall, there are a number of limitations that slow down in-process related decisions. Now, the ELA platform overcomes many of these limitations faced by the ELISA, and these points are highlighted here on this slide. So they include greater dynamic range, high throughput, and greater precision. And over the next few slides, I will demonstrate the benefits of the ELA platform over the ELISA using real-life process development examples. So one of the challenges in process development is that there is a broad range of capsid concentrations across each unit operation. So you can see in the schematics to the left that the range consists of 10 to the 10 to 10 to the 15 across the entire workflow. And often we're changing parameters to improve titer and recovery throughout the whole process. So what this means is that usually there's low confidence in the concentration of a given sample. Now, because the ELA has such a higher dynamic range when compared to the ELISA, and you can see this here with the graph in the middle. So you can see the ELA is shown in blue and the ELISA in orange. The broad dynamic range means that concentration uncertainty of your sample becomes less of a problem as you're targeting a much broader range now compared to the ELISA. So here I'm showing two different sample types, so crude samples and purified samples, and three different samples were taken of each of these sample types. And this was all run on one ELA cartridge. and actually using only one dilution across all the samples. And on this cartridge, we were able to capture both crude samples, which have a much lower capsid per mil concentration compared to purified samples. This means that we can go in with fewer dilutions and be really confident that even with these fewer dilutions, we're going to target the concentration of our sample. So overall, running fewer dilutions and allowing us to make decisions across a broader range of process samples is really adding value to our process related decisions. So, as I mentioned earlier, a challenge in process development is the vast number of analytical samples. And so, throughput is a critical parameter to making informed process decisions. So, we often run AMBER 250 miniature bioreactors. And you actually can see a picture taken from within our lab of the AMBER 250 bioreactors on the left there. And we often purify these bioreactors to assess changes upstream has on downstream. So, for example, we might want to assess changes different culture media has on the process workflow. So, we typically compare the titer of 6 bioreactors and assess recovery across the process workflow. So, I'm showing this as a schematic there on the right of the slide. So, with this sort of experiment, we generally typically generate around 30 samples, and you can see where these samples are generated across the workflow on the slide. So to analyze these samples on the ELISA, we would have to run multiple plates. And in fact, you can see there that we'd have to run 3 ELISA plates. So what this does is introduce plate variability, which we know from experiments within our lab that this variability ranges from 10 to 20%. Now, if we were running these samples on the Ella cartridge, because it has such a higher throughput compared to the ELISA, we would actually only require 1 Ella cartridge. And the schematic there is of a typical plate map we would generate for the Ella cartridge. So running all the samples on this one cartridge means we completely eliminate any influence of inter-assay variability. So this again is allowing us to make more informed decisions of our process development workflow and samples, and also allowing us to create a smart design of the workflow. So the final case study I'd like to go through is looking at the whole process. So for example, here in the slide, I'm using 50-liter bioreactor and looking at upstream all the way to downstream, and looking specifically at this slide of running multiple batches. So there's several reasons why in process development you may require to run batches. And the example I'll use is, for example, you want to look at the difference between 4 different constructs, and you want to look at this difference across the entire workflow. So if you were to do this, you would typically generate 13 samples per batch and times that by 4 for each batch. So this means you'll have a large number of analytical samples. And actually, when you figure out how many ELISA plates that requires, it's 4 ELISA plates compared to 1 Ella cartridge. So therefore, as mentioned previously in the previous slides, again, we're removing inter-assay variability. So not just is it increasing our precision, but also our efficiency to make process-related decisions. So in conclusion, process development leads to a generation of a large number of samples. And one of the key challenges is throughput and precision. And the Ella platform has really been instrumental to increasing our overall throughput, and by doing so, allowing us to make more efficient and accurate process-related decisions. So thank you for your attention.